Welcome to RX, How I Got There. I am your host, Ebere Iboko. In this podcast, we profile and tell the stories of pharmacists and others doing interesting and inspiring things with their careers, with the hopes of showcasing the different opportunities within pharmacy. If you're a student or a pharmacy professional, this podcast will help to broaden your career aspirations and your worldview. Our guest today is Dr. Peter Atadja, Chief Scientist Officer at a biotech startup company in Boston, Massachusetts. Peter is a world leader in drug discovery and a pioneer in epigenetics. In this episode, you will be thrilled by his passion for science and his humility. There is so much everyone could learn from Dr. Peter Atadja. Hi, and welcome to RX, How I Got There. This is a video podcast where we try to tell the stories of pharmacists and people who impact pharmacy, with the hopes of sharing information that could be useful, enlightening, or even inspiring to pharmacy students and other pharmacy professionals. When I started this podcast, it was in response to pharmacy students in Nigeria who were curious about how pharmacy is practiced outside Nigeria. Since then, I have found that even beyond the students in Nigeria, there are students everywhere and other professionals who are curious to hear about best practices and the careers of pharmacists around the world who are doing very interesting and, in some cases, non-traditional things. Today's episode is one I'm very excited about because I'll be speaking to someone who I consider a friend, Dr. Peter Atadja. But even more than that, Peter's background is really fascinating. He was born in Ghana, where he got his pharmacy degree. Then he moved to Europe for a second degree and a final degree from North America. I met Peter in Shanghai, China, over 10 years ago when we were both working there. He's a scientist that led a discovery unit for a multinational company, and he'll tell you about his achievements, which include the development and the innovator that is a patent holder for a groundbreaking cancer treatment. I look forward to hearing from Peter. I'm sure that there's a lot to learn from his career, what drives him, the decisions he's made over the years. And by the way, in addition to all that Peter is doing, he's spent some time now back in Ghana teaching and mentoring young students. So I don't know all the places Peter and I will take this conversation, but I bet there will be a nugget for anyone who is listening and who is interested. So stay tuned and welcome to RX, How I Got There. It is so exciting for me to welcome Dr. Peter Atadja to this video podcast, RX, How I Got There. In a minute, I'll invite Peter to tell us about himself, but it is fascinating that I'm talking to Peter while he is in Accra, I believe, or is it somewhere else in Ghana?
Yes, in Accra.
Okay, so Peter is in Accra, Ghana, and so far all the technology is working, and we hope it will stay that way. But Peter, welcome. I always like to start on a personal note. From your LinkedIn profile, I read that you obtained your pharmacy degree from the Kwame and Nkrumah University of Science and Technology in Kumasi. And then you went to the Hebrew University of Jerusalem, where I believe you got a master's degree.
Yes.
And then further, you went to the University of Calgary for a PhD.
Yes.
That is truly global. So it's a pleasure to have someone with that breadth of experience. Do you mind telling us a little bit about yourself, including all the things you're doing now?
Okay. So thank you so much, Auntie Berry, as I call you. And it's good to have this conversation with you. For us, the more reserved ones, we don't give ourselves the opportunity to actually tell about our life journey until someone like my dear auntie, to whom I cannot say no, asked me. And I think that together we'll be able to bring out something that will be valuable and helpful to those following us. So thank you so much, Auntie Berry, for bringing us together. And also, I still remember the very first day that we met in Shanghai, China, in a lineup to the Concordia School office. And they were all expatriates, but all of them were Caucasians and others. And then all of a sudden, my wife Cynthia jumped on you as if you were a long lost friend or a sister. And from then, you've been from that time, we've been very, very close, and we've shared each other's professional and also personal stories. Thank you so much also for being a very good auntie to my girls. Thank you. So yes, I graduated from the Kwame Nkrumah University of Science and Technology with the Bachelor of Pharmacy. But I think my story with drugs started way before then. You know, my father, who was a traditional ruler in the 1940s, was trained as a teacher. But when the opportunity came that the colonial masters wanted to train people that would work in clinics, and although not full doctors, but can work to see patients, he changed his mind and trained to become, sometimes people call him doctor, others call him dispenser, others call him health superintendent. And so, long story short, he saw patients, and he became very passionate about public health, and he started actually establishing rural health clinics all over, our area, and so he worked out of those. And it was that time that I actually really became acquainted in my very early ages, 5, 6, very acquainted with medicines. And the name, actually how medicines are called in my language, literally means the root of a tree, atike, a tree root. So actually, I was very curious. And when I was little, about 6, I thought that then maybe all drugs came from the root of a tree. So in my grandmother's compound, he has an orange tree that I dug out the root one day, peeled it and ground it and tried to mold a peel out of it. That is how my very first memories of trying to get myself to make a drug came to me. Fast forward, I was a very good chemistry student, and also liked a lot to learn a lot about the human body. So after high school, actually, I applied to a number of schools and applied to both pharmacy and medicine. And so I applied to the Kwame Nkrumah University of Science and Technology, which had the only pharmacy school at that time in Ghana. And a medical school, applied to the medical school as well, and applied to the University of Ghana, Legon, which had the only other medical school. In the University of Ghana, I was offered dentistry. And then at the University of Science and Technology, I was offered both medicine and pharmacy. Guess what? When I was leaving home, everybody knew that I was going to do medicine. And then when I went, I was roomed with one of my friends at high school, who was then a second-year pharmacy student. And when I got into the room with him, he started telling me about, how exciting pharmacy is, and knowing that I like chemistry, I'd rather enjoy pharmacy. Not even to think that there was a very modern pharmacy building on campus. I followed my friend and registered for pharmacy instead of medicine. And that's how my life as a pharmacist, pharmacy student started. During my studies, I was kind of a rebel student. I really did not like, lecturers and professors dictating notes to us, although they did that because at the time it was very difficult to come up to, get the up-to-date books and journals. So they will make the notes and come in. But I really, hated that. So I wouldn't copy any notes. I would only listen and then go to the library later on, and whether it was the latest version of the book or not, I would read the book, I would read the journals, and that is how I also begin to pick up how things came to being, because I like to read the stories about, how viruses were discovered, how the antibiotics were discovered, and things like that. And that is how that curiosity led me into drug discovery.
That's a fantastic story. I hadn't realized what way back your interest goes. And of course, thank you for reminding me of how we met, because in Shanghai, in the sea of, as you said, Caucasians and Asians, meeting you and your family, I knew instantly that I had met my sister and my brother and your three lovely daughters. So that was, fond memory, going back to fond memory lane.
Yes. And I hope I will be able to get the opportunity to tell you more about how my real research life and drug discovery life, went from here.
Absolutely, because that's where we're going into now. You mentioned drug discovery. Yes. What is discovery research? I know that in Shanghai, you worked for a multinational company, Novartis, and you actually led the discovery research team at Novartis. And as you well know, a huge part of our audience are pharmacy students in Nigeria, in Ghana, and elsewhere. So for those who don't know, how would you describe discovery research? And also, as a pharmacist, how
did I get there?
Yes, how do you get there?
Okay. Okay, so currently for me, and for anybody that would listen, I always describe discovery as just going along in my rich father's garden and looking under the right stones to find solutions for problems, especially problems of health and disease to develop therapeutic solutions. But yes, I mean, what is discovery? And the discovery is actually, I mean, picking picking a problem, a health problem, a medical problem, trying to understand, doing everything to understand the very deep details of as much as one can know of that disease process, right from the molecular level through cellular level, all the reactions and actions within the human cell, right into the organs, and then how they affect the biology of the individual and the morphology of the individual, translating into the well-being of the individual. So knowing exactly what is normal or what can be described as normal. And then now that you have learned and understand what is normal, then also having an eye of when normal veers away to become abnormal, identifying the point or the reasons why that abnormality happens. And then when you identify that which is causing the abnormality, which now we would call a target, then you try to identify what one would call the Achilles heels of that target. You try to understand, and the target could be a process, could be a protein, could be a multi-protein complex, could be even an organ, and how the organ is arranged within the human body. You find out the Achilles heels of the process that makes it become abnormal. And then once you identify that point that you can attack, then you now go back to your lab and now try to mold or try to create a molecule. A molecule could be a small molecule, larger molecule like peptides, proteins, antibodies, and shape them in such a way that it can interact with that protein or that target within the human body, and tackle it in such a way that it attacks that and nothing else that resembles it, to selectively, interrogate that, either activate it if it is not working well, or inhibit it, overactive, and then translate that into another process that will then ultimately affect the biology of the individual and the health of the individual. So as you can see, drug discovery is a multidisciplinary process. I mean, going from even human pathology, cell biology, molecular biology, biochemistry, chemistry, and all the others that you can bring together to bear to really interrogate the processes in the human body so as to either reverse or bring back activity that is not working. And whilst doing that, you have to also find ways of monitoring the exposure of the human body to the foreign molecule that you have put in the patient, to make sure that it is within the right level and for the right duration, and be able to communicate and work with clinicians to understand it enough so that you can test it in the sick patient and find ways to test it in one patient as well as in the population of patients, to know what data that you have is significant, or it may just happen by accident in one patient. It's such an exciting field that you are always feeling like a kid in a candy store.
That's exactly how you sound. Your passion is palpable. So, as you just said, it is multidisciplinary, chemists, biologists, bioinformatics. As a pharmacist, I know you led your group.
Yes.
What do you feel equipped you to play that role?
Thank you. First of all, as a pharmacist, in my pharmacy education, I got the opportunity to tackle all these various aspects, right from the physiology, anatomy, biochemistry, microbiology, chemistry, medicinal chemistry, formulations, pharmacokinetics. We did all, as you know, in our Bachelor of Pharmacy. So that's actually nothing that I interacted with during my drug discovery. Nothing that we were doing was new to me. So that was something that really helped. The other thing that helped is that I am able to work on whatever discovery problem that I'm doing. I am able to work on it with the end in mind. So I work with a number of scientists trained from Harvard, Oxford, wherever, and they are the experts in their disciplines and what they are doing. But I am the one who actually have the context of what a drug meant. And working with that end, in mind, it gave me a more a comprehensive and holistic impetus to be able to do what I'm doing, knowing where I am going. Most molecular scientists and things like that have only dealt with animal models. I had dealt with the human patient, and I understand a lot about the human patient, and I understand a lot about therapeutics. The pharmacist and his or her training becomes the expert, the best expert in drugs. So if I am, working on a problem that will become a drug, I'm working with the end in mind. And that is, I think that of everything that my pharmaceutical training gives me, being able to work, knowing the end in mind really is helpful.
That is the best way I've ever heard it described. So thank you. I don't know about schools in Ghana, but pharmacy schools in Nigeria, in our final year, our senior year, we are required to do a project. And it can be from pharmaceutics, pharmacology, or any of the departments. And that's an experiment where you learn the steps and then you write a thesis, you write up your project. Do you mind summarizing with this backdrop, maybe using drug discovery? Because when you mention the end in mind, it is really interesting. Drug discovery, as I understand it, is at the beginning, but having the end in mind makes you even more successful. So do you mind tracing for us just the steps to go from the beginning to the end in either drug development or however you see it?
Okay, thank you. Thank you so much. And I think that I just have to be tickled, and I'll tell you everything. And thank you for giving me the opportunity. Yes, we at the Nkrumah University of Science and Technology also had to do not just not a dissertation, but also a research project. And my specialization was more in medicinal chemistry, at the undergraduate level. And my project had to do with isolating some specific analgesic alkaloids, non-morphine type alkaloids from some herbal, from natural seeds, and then characterize them. That was what I did actually. And when I graduated, my medicinal chemistry professor, who was also the dean, got so much interested in what I was doing that he decided that I would, everybody was going for internships in hospital and things like that. He decided that I would stay behind with him in the lab to continue to be both a teaching and research assistant. And it was this that I was doing one day, and we were sitting down. This is Professor Juma Bedu, who had just returned from University of Lagos to set up the pharmacy school at University of Lagos, and came back, and he was saying that there is one thing that really bothers him, and talking about finding a problem and devising a solution, and also the end in mind. And him being a pharmacist, he said morphine and codeine had the exact same structure, except that codeine is an acetylated version of morphine. However, with that acetylation, codeine picks up an antitussive activity, which is suppressing the cough center, which, morphine doesn't do well. And morphine, lacking that acetyl group, also is more addictive. So how is it that one can use this difference, just these acetyl groups, to really make a better morphine and codeine, something that is, better than both morphine and codeine? So that became he and I would sit down and be designing molecules and thinking about the way to do this. I told you that, I always went to the library. I got to the library, and there was something that we were taught in pharmacology when they were teaching us psychoactive compounds, and it was about cannabis and how cannabis produces extrasensory perception, where sometimes people who take cannabis or smoke cannabis even think that they are flying, and so on and so forth. So that really, was something also that was bothering me. What in cannabis can really cause that end in mind that we're talking about? I was in the library, and then lo and behold, I was reading a Scientific American, and saw that an Israeli scientist called Professor Meshulam had just identified and isolated a compound called tetrahydrocannabinol, which is the active ingredient in cannabis. I could not contain myself. I tore my notepad, wrote a letter with my pen, folded it, put it in an envelope, went to the post office, the university post office. I didn't even know how much it cost to mail a letter to Israel, because at that time Ghana and Israel didn't have a relationship. So I just bought stamp, put it in, and then sent it to Israel, and told Professor Meshulam that, I wanted to know more about this, because I was also thinking about morphine and codeine and how they have, these central properties, addictive versus antitussive, etc. So I wanted to know more about THC. And did he have a place in his lab to do it?
Wow.
And then I sent it 2 months later. I didn't hear anything. And then another opportunity came up where the International Pharmaceutical Students Federation was, sent an opportunity for someone to be nominated to go and do a 1-month internship. Guess where? Either in Israel or in Holland. I said, send me to Israel. So that is how I got to Israel for a 1-month internship. Went, and then as soon as I got there, I asked, where is the Hebrew University? And they showed me how to get there. I got there in the whole big campus. I started looking for Professor Meshulam's lab. Finally got there, and I went, and then I met him. And long story short, I mean, I finally became a graduate student in Israel, and was able to get into this area of drug discovery. In fact, although I did not finally work in Professor Meshulam's lab because of, I mean, funding and things like that, the lab that he took me to and vouched for me that based on what he thought I knew, I mean, I could be very successful working there. The day I got there was the day that that professor told me that an organization called Sibagaigi in Switzerland had just licensed a compound from their lab, which was an anticholinesterase compound to help with the symptoms of Alzheimer's. And so I said, from this lab to
Sibagaigi in Switzerland.
Wow, that means I could do it too. And that is how I got initiated, and I got immersed, and I never looked back. And we can talk about the milestones and the achievements later.
Wow. So I want to just make an observation, because when I've spoken to students, especially those in the sciences, the core sciences versus the social sciences, the steps of doing research sometimes are not always clear and distinct in their mind. But you know, you've mentioned more than once going to the library.
Yes.
And I tell students, your literature review is where you start.
Yes.
Because that's how you know where others, the questions that are yet to be answered. And you build on what people, the body of knowledge.
Yes.
And so for you, you know, with the first step was all the library work you did to even crystallize the question. And in so doing, putting it in another context, you maybe highlighted the hypothesis, so what one might expect, and then went on to design experiments to prove or disprove what one might expect. And I know sometimes the steps blend into one another, but in my mind, that's a basic understanding, especially for people doing it for the first time. So I just wanted to make sure that, you know, someone as successful in the sciences as you agree that those rudimentary steps are still necessary. Yes.
Okay. So you talked about the sciences and even blended in social, sciences. And I tell you, being curious about your environment and what is happening around you is where it all starts. The curiosity to identify a problem. And that problem could be a social problem that in the long run may have a scientific solution. So that curiosity to identify a problem is first step. Then if you are curious about the problems around you, you would start reading in that area and start reading about what others have done in that area. And as you read it to educate yourself, your curiosity will be asking you, oh, what about this? What about that? And even challenging some of the assumptions that were being, others were doing. However, when you then get into a specific work that someone has done, start from the introduction. The introduction is the one that gives you the best benefit. It tells you the broad question, the broad problem. Could be, a social. Why are Africans not, being affected so much by COVID, and so on? So, what the broad questions is, and what other people have done, and what they have got, and what questions, what specific questions in specific areas have they asked? Is it genetic? Is it environmental? Is it prior exposure to broad? I'm talking about the COVID issue. Is it a prior exposure to other broad things that have insults in their body that have already set up their immunity? What then? Now you are getting now into am I talking about immunology? Am I talking about public health? Am I talking about pathology, etc.? Now you begin to read and then find out what questions did people ask, what answers did they get, what remains to be answered, or even what experiments did they do that quote failed, that eliminates for you what you shouldn't be doing. And now when you come to what remains to be done, then you ask yourself, how would I contribute to this body of knowledge? Do I have any systems or models to be able to test what I want to do in? And if I have it, do I have the reagents and to be able to bring it together, and the equipment and tools to be able to do it? And would I know if I got the right answer or not? And if I answer the question, am I fully answering or only partially or not? And then how do I collect and analyze data? And how do I interpret the data that I have analyzed in one system versus multiple systems, to know whether it is significant and I can make a broad statement? And how do I enable the field? How do I communicate what I have with to put into the context of what is already happening, and so that everybody else can understand, challenge me, or do something else? So yes, it starts from curiosity, being curious and interested in problems, asking questions, educating yourself what has been done, and what exactly were the questions or the hypotheses people were testing, what results did they get, what remained to be seen. Getting a model or system to do it, doing it, interpreting your results, and then communicating it very well to advance the field.
Great, thank you. And I know you're a pioneer. You've been described and cited as a pioneer in epigenetics.
Yes.
And you were talking about science and how the body is impacted by what's happening around it. For those of us who are not experts, what is epigenetics?
Okay, so first of all, let me tell you what it is not.
Okay.
Because, most of us have a good understanding of genetics and how genetics really helps the expression of traits, phenotypes, etc., etc. And in that genetical framework, when you have mutations in which are, abnormal sequences of a specific gene, what it translates to. Now, in the area of epigenetics, you may have the right genetic sequence, gene sequence, the right DNA sequence, but then you may lack the traits that the sequence entails because that sequence is not expressed into the protein, which is going to activate the effect in the human body. So the epigenetics, that is beyond genetics or above genetics, is those regulatory processes which make, the gene sequence being there, but which regulates its expression or silencing to enable an effect to happen. So epigenetics processes are those that interact with the genetic sequence, DNA sequence, to either regulate the expression and translation of the sequence into an effective molecule like a protein to work. It is these mechanisms that regulate the expression that is epigenetics. And yes, I mean, I have been referred to as a pioneer in epigenetics, and this is why. Still in Israel, I went to an International Union of Biochemistry that was held, I think, in 1988 in Jerusalem. And here was, Professor Robert Tijan from Berkeley, who was giving a presentation on how genes, the expression of genes are regulated. You know, you may have the gene sequence and then it is transcribed into mRNA. And so how the RNA polymerase regulates the transcription of the gene sequence in the mRNA to be translated into a protein. Again, it was something that, I mean, my mind couldn't contain. What it is possible to really regulate how genes are expressed. And this is what I told myself. Then it means one can identify what a gene switch is. So you can switch a gene off and switch a gene on. That is what I wanted to learn in my doctorate, for my doctorate degree. And that is where I started writing to universities that I wanted to understand the regulation of gene expression. And Calgary was one of the first that accepted me. And I just, and I went there. And what I started learning there was my project was around the aging process. And here was a young professor who had just trained from Cold Spring Harbor Laboratory in New York, and came and was establishing his research. He always called me his best student, although I was his only student. And he wanted to understand the mechanisms that regulate the aging process. And there is an aspect of cell growth that, he had a model of. And when cells divide, every a cell in our body has only a certain number of times that the cell can divide and then stop. You cannot divide, you are a lung fibroblast, you cannot divide more than 72 times. You have to stop, dividing. And what he was able to identify was that the number of replicative ability was proportional to the age. So if I got cells from a mouse, they replicate maybe shorter times than from a monkey and then from a human. So he was able to relate that to longevity and aging process. But it also got us to understand that cancer then could have been linked to that, because cancer is the continuous proliferation of cells when they should not proliferate. So my exciting this thing was to understand, what were the factors that regulate the replicative senescence, as we call it, and how it relates to cancer. Long story short, I identified something that was called tumor suppressors, and then realized that actually there was a genetically program into the cell to cause it to stop replicating, and if it does, then it will become cancer. And this is how some tumor suppressors like p53 might act on the cell cycle. That was one of my biggest publications, because the director of Cold Spring Harbor Laboratories was Jim Watson, the one who solved the DNA structure with Professor Craig. And he was so excited that I got this actually aspect of gene regulation that he decided to be the one that communicated this in the proceedings of the National Academy of Sciences, which was published, I think, 1996. So that, this is how trying to know how genes were regulated, got me into this, epigenetics. After my PhD, I went to the University of Toronto at the Princess Margaret Hospital to understand gene expression mechanisms in Burkitt's lymphoma. And in Burkitt's lymphoma, there was this, there's this gene called MYC, and it was MYC that really was really driving the disease. It was this thing that I was doing that one day I got a call from somewhere in the lab. I took the call, and then there was somebody on the other end saying, okay, I am the director of research, a woman, I'm the director of research for a newly formed company called Novartis. And Novartis had then become merged. Was a product of the merger of what? Ciba-Geigy and Sandoz. And we listened to you present at the American Association of Cancer Research, and we liked, the way you talked about the regulatory of the cell cycle and how it might help in tumor suppressor mechanisms to be able to target for cancer. So would you like to consider a position with us? And I said, yes. And then they said, the person said, full circle. Then the woman on the other end said, okay, we also found out about you that you went to school in Israel. Do you speak Hebrew? And I said, yes. Then she changed it into Hebrew and started asking me questions in Hebrew. And I responded. When I responded, she said, as soon as you put the phone down, go to the Toronto Pearson Airport. By the time you get there, go to the Delta counter. There will be a ticket for you over there. Take it, fly over to New Jersey, and then we'll talk. And that's how I got into the field of targeting epigenetics, which really, remember the gene switch thing that came to me. Now I want to get molecules that did it, and they gave me the opportunity. No one was doing it. I was so young and naive, nothing could go wrong. I went for it. And by God's grace, things went one after the other, not without challenges. Went one after the other. And thank God, my group's work became the first epigenetic compound that went in for clinical trials. And then others went in and were able to move it right up to FDA approval and global, I mean, EMA, Japanese and global approval.
Wow. And I know it's no small feat. In fact, I want to make sure that I acknowledge that I became aware that you, my friend and brother, led the development of the first drug, Panobinostat, based on epigenetics for relapsed refractory multiple myeloma, which is a type of cancer.
Yes.
And you led the development, like you said, all the way to global approval.
Yes.
What an achievement.
Thank you.
So,
thank you. And just coming from KNUST.
I know, but you know, that's driven by passion.
Yes.
Willing to work hard with all the curiosity, not afraid to ask questions and to make connections with people all over the world. See how far it can get you. I think it's such an inspiration to anyone who is listening to us, especially any young students in some university where we all come from in Africa who thinks they are stuck where they are. I mean, look at you, you know, and what you have contributed to science and to the world. So tell us a little bit about that experience. Like you referenced, it wasn't without challenges.
Yes.
Any highlights of some of the highs or some of the lows?
Yes. So, of course, the highs, I can talk about them till the cows go home, or come home. But the final thing is that we got an FDA and EMA approved a drug for multiple myeloma. But I'll reserve one of the highs and tell you later. But the lows, yes. I mean, I came to Novartis and I started this project, but this is a project that, I mean, not many people are aware of the system and really care about. Here's a young scientist that is starting his work. And it really did not draw a lot of attention until when we did a screen that we got a hit in the screen with a drug that already we knew had anti-cancer properties, but which had not been developed into the clinic to hit in the mechanism that I was testing. Now, the mechanism that I was testing was called the chromatin histone acetylation process. And anything that targeted histones, you know, we all know that histones were the so-called packaging material of DNA. So the challenge that I had from all over is that how can you target something as pleiotropic as histones to think that you can get an efficacious drug out of it without killing the patient? And so there was a lot of pessimism on the work from inside and outside. And what did I have to lose? So I just kept working to prove. I was open to the challenges. Everyone's challenges would come, and I found ways to design experiments to prove whether what they were saying was right or was wrong, because I also don't want to be barking the wrong tree. And I then identified that there's a specific modification in a histone that actually is affecting the whole process. And if I can tackle that specific modification, the whole histone will still be there. But it was the overactivity of that which which was causing it. So I tried and then got molecules that will, interrogate that specific activity. And lo and behold, I was able to show that, this drug that I had was able to kill cancer cells, but not normal cells. And that was, hooray, golden goose of that of that experiment. And that was when people started paying attention. And then at that point, I had to change, they had to change boss, I had to change bosses, I had to change supervisors, which is another major, a challenge in research, because this research work take like over 20 years. Now, another supervisor comes with their own interests and with their own priorities, and before you realize it, you are assigned other things. But I kept working on my stuff, even whilst getting my day job. The one that I will get paid for. I started doing it until I got another, result where, one of the compounds was able to shrink tumors in mice in a dose dependent manner. That was when we put together the whole program, and then now moved the program into to get a drug candidate through optimization to get a drug candidate, and moved that drug candidate into the clinic. We got into the clinic, and then this drug was first being tested in AML, in leukemia. It was actually showing efficacy in leukemia. And then we got a call from Germany saying that, oh, the drug actually, because one of the patients had what is called Torsades de Pointes, which is a heart issue. And another challenge. So the FDA had to put this development of the drug on hold. Now we have to understand why the drug was causing Torsades de Pointes, and what we needed to do. A long story short, we identified that this drug was causing prolongation of the QT, was causing QT prolongation, and that we could not continue developing this drug. So we went back to the lab to try to synthesize, redesign a compound that would not prolong the cardiac QT, and moved that. So the first one was the very first epigenetic drug to get in the clinic because the efficacy, but you had, QT prolongation. And then we brought in the second one. By then, we found, other ways in which this drug might work preclinically. So, and mind you, we're testing this drug all over the world. Then in Australia, there was this woman who had a type of skin cancer called cutaneous T-cell lymphoma, and she had failed about 7 lines of therapy. But and this woman was facing death, and then her granddaughter was going to get married middle of the next year. It was around Christmas. So she begged the doctors, get me something that can take me just to June, and we will and so that I can see my granddaughter get married before I finally let go. So they gave her this drug, Panobinostat, which we called LBH 589, and I have the pictures. 3 months later, this, 80-something-year-old woman, the skin has become like a baby. And that was when we knew that we had a drug. But for me, I was looking at that grandma being able to hold her granddaughter to the altar. This is the changes. This is the impact that we can have in people's lives. Full circle back to the social, of the human experience, and that is where, we got, that woman got to live a lot longer. I unfortunately, I wasn't able to meet her face to face, but I was able to talk to her at that time on a phone conference. So the challenges, and then the challenges of not being believed, being given other things to do to distract you, but you kept doing it. Your boss is changing, the side effects, and then coming back to reshape it, and then taking the wins when you can get them.
How amazing. So many nuggets, so many lessons in that story that ends in triumph. Wow. That's so amazing. And like you said, the human aspect makes it all the more fulfilling that, despite the lows, the highs that impact human lives make it worth it.
Yes.
How encouraging. Thank you for sharing that.
Thank you.
I know that in addition to everything you've done, you have maintained interest in academia. Do you mind telling us a little bit about your academic pursuits, whether they are sometimes on the front burner, sometimes on the back burner? And I know that I'm talking to you while you're in Ghana.
Yes.
And in Ghana, you are an adjunct or full professor somewhere. What is your interest there, and how do you combine it with all these many strong passions that you have?
Yeah. So first of all, you and I were in Shanghai when Ebola was happening in West Africa. And although we were working in the pharmaceutical industry, we felt helpless. We felt kind of hopeless. And I know many times we really talk about, what is going on. And it was at that time that I said, I wish that we had the capacity and the capability to be able to understand the diseases that plague us. Otherwise, unless it affects others, unless it can, a drug discovery is also something that is around people who put in investment. So they have to get things that work for them. That was the time that I came home one day, and I was talking to one of my previous classmates, actually, university mates, who was a doctor and was the dean of the School of Public Health at the University of Ghana, Legon, and was being asked by the government to go and start a brand new university in my home region, in the Volta region. So Prof. Abinka invited me and said, Peter, I know you from when we were students, and I know your passion as well. And I want you to come and help me establish the foundation school called the School of Basic and Biomedical Sciences, which would found the scientific basis of all the professions, nursing, medicine, pharmacy, etc. So I agreed, and in 2016, I packed bag and baggage, as you knew, came down, and I was appointed the dean of the School of Basic and Biomedical Sciences here, and established a program that, first of all, helped recruit a lot of young faculty, and also faculty from other universities to really build this program to set the basic biomedical foundation for the professional for the health professions. That was done between 2016 and 2017. I also leveraged my international relationships to bring in equipment and some funding to really start biomedical research in this place. And I mean, one of the research that I actually started was in how the human intestinal microbiome affects the sickle cell patients. And coming full circle, it is something that I'm working on right now in a biotech startup elsewhere. So that is, I mean, what I did was this that we were doing, that the university also decided to establish a pharmacy school, a PharmD pharmacy school. And one of my other previous colleagues from University of Science and Technology, who actually came to become the pro-vice chancellor. I mean, we got together, put together ideas and curriculum, and he had actually established the PharmD. He had just established the PharmD at KNUST. We put that together and brought in the very first PharmD students in 2017 at this a new university, before I returned back to Novartis, but continued to hold a full professorship there and coming back. I mean, periodically, I actually got some established some relationships with Novartis where they interacted with the biomedical research on sickle cell, which I mean evolved actually into a national program right now, where Novartis and Ghana has a collaboration in sickle cell. So I keep, as you rightly noted, I'm currently still in I'm in Ghana now. Because of COVID, we are able to actually work remotely, so I'm able to come back more frequently. I mean, suffice it also to say that the biomedical laboratories that I helped started has now become the national COVID testing laboratories for the eastern flank of Ghana, where you have the OT and the Volta regions and part of the eastern part of the eastern region. And of course, some Togolese also come. So, I mean, things that you do without realizing, create the capability, create the capacity, and then, I mean, when the challenges come, when the problems come, you are there. And we are also developing both the faculty and graduate students right now that hopefully will be able to take over from us.
Wow, I thought I knew what you were up to, but just this last bit you said, I have so many thoughts, some of which I will follow up after we are done with this. But first of all, sickle cell, to be doing something on sickle cell that's getting international attention and investment, that's wonderful for all the patients that you and I know exist among the Black race. Then secondly, the impetus for my doing this, and for you as well, has been targeting those who are coming behind us, the young ones, the students, the professionals. But actually, in this few minutes of what you said, to me, you also model for people like me, people who are looking for how to give back. Because with your international relationships and your expertise, you are connecting people at Novartis, building capacity, in Ghana, sourcing equipment, like you referenced. It is fascinating, all we can learn, all the lessons that different groups of people can learn from your own story. So I really appreciate that. I know we haven't mentioned anything about the time in Shanghai, but I'm also very conscious of how much time I've already taken. So I kind
we can do the same thing. Yeah, go ahead.
Wind down by asking you about the lessons you wish to share. And please feel free to draw upon lessons from China, your time there, the US. You now go back and forth. You reference some things you're doing with biotech. I mean, you are juggling so many things that one hour can hardly cover all the exciting things you're doing. But just in closing, please feel free and take as much time as you have, because I have time, but I'm conscious of your own time, to share lessons.
Yes. So talking about lessons, yes, the lessons abound. But I think that the biggest thing that one needs to have is the humility to know that, one, I don't know it all, and two, there is a lot I can learn. And that the diversity of knowledge, when aggregated and integrated, produces much better than what one aspect can do. And I can just tell you some of the few things that I have learned working internationally, really all over the world. And starting from the United States. United States is a country that funds research at the highest possible level. So once you can dream it, and once you can get the funding for it, you can do it. And there is nothing that beyond the imagination that you couldn't try to do in the United States. And that really helps. That can-do attitude helps the whole research ecosystem, brings all, lifts all up. The ecosystem is such that it's very interactive, it's very collaborative. Teamwork is of prime importance. And as I said, research and discovery, especially as it is multidisciplinary and all the disciplines are there, you can bring it together and you just have to harvest it. And if you are humble and know that and teachable, you can get and learn a lot. Both the depth and the breadth is there for harvesting in the United States. And just, if you have the opportunity to go there, dig deep and drink deep. You can't imagine how much you can learn and how much others can learn from you. They also actually take particular attention to the fundamentals of things. There are universities that don't even do any professional training at all. Some of them are called liberal arts universities. But some of these liberal arts universities that focus just on the fundamentals and the philosophy of education produce some of the biggest scientists, because they focus on the fundamentals. So in the US, the fundamentals matter, first principles matter, and they dig deep and they dig broad. So that is one part. Then you go to China, and China is when you look at that, you and I were in China at one of the most accelerated development times, and China, most of its a real innovative development is about just 30, and in the last 30 or so years. So, but they want to catch up. And one thing that I saw with my Chinese colleagues, try again. If at first you don't succeed, try again. Keep trying. If you have to do it multiple times to be able to get it to work, they will do it. And that is the kind of persistence and perseverance of, repeating until I can get it. And that gets them to hone their skills in doing it. And also one thing that I saw is that the Chinese fingers actually can do finer things than, they're able to do very delicate, things, thread the needle so delicately, and it's a strength that they have. And if one is able to get that in addition to into other strengths from somewhere, I think that will work very much. And
sorry, may I interrupt you to make sure that what you're saying is clear? Are you saying that in China, you found scientists to be particularly meticulous?
Yes.
Or, okay.
Yes, meticulous, attention to detail, and types of animal surgeries, things that the surgeries that they do under the microscope that, I'm asking myself, how did they do it? Yes, that I also found, and of course the ability to repeat things until they get it. And in China, and I say this in a positive way, we don't mind to copy. We don't mind if we don't want to, start from the scratch and reinvent the wheel. Look, I not iPhone, the other I just forgotten the name. No, it's it's a China model that they build on a platform such as iPhone, and Xiaomi. So you don't have to reinvent the wheel. That is something also that I learned, especially for Africa, when we don't have much time. We can do things that helps us to leapfrog. And then when we leapfrog, then we will be able to, solve some of our problems. And that's China. And then in Israel, I went to Israel at a time when it was facing a lot of security problems. It was not, it didn't have a lot of interactions with the world globally, and so they were not as rich as they were at this time. But knowing that, they intentionally choose not to be jack of all trades. They identify where they can be the best in, and then they work hard on that. If it is not the best, don't try it in it, or don't attach it to an Israeli scientist. They identify where they can, and then they drive that hard. And I learned that you don't have to be everything. You just have to contribute a critical aspect to the global science, so that things can move on, and then people cannot help but fund what you are doing because you are critical. And that is what the Israelis are doing. And when they do it, they don't, they know that they want to get to the end, turning the desert into a fertile place where Israel is feeding Europe with their agriculture right now, is not for the faint-hearted, but they chose that and they dug deep into that. Today, at NASDAQ, in specific areas of artificial intelligence, in aerospace industries, in certain aspects of biotechnology and things like that. Today at NASDAQ, it is the Israeli companies that are making the biggest, moves on the NASDAQ, but they chose specific areas and drove hard into it. That is also something that I learned. Now, you don't have to be the jack of all trades, but try and be the best at where you are. So this is just few examples from the United States, from China, from Israel, and for all my my compatriots out there. Everything that I said about the perspective from each of the places, I said it with the most positive, perspective, and nothing has a negative connotation to it. I learned so much from all you guys, wherever I interacted with you, and that is what in aggregate makes me what by God's grace I've been able to do. Thank you.
Thank you so much. This is so rich, so inspiring, so enlightening. And even for me, who have known you for over 10 years, I'm so proud to be your friend and sister. I have learned a lot even in this conversation. I have ideas of how I want to be do more. And I hope that everyone who is listening to us, your enthusiasm is infectious. Your passion about your work is admirable. So I'm really grateful that we had this time. I'm thankful that our technology allowed us, despite the distance, to have this rich conversation. I look forward to following up on all the things you're doing and cheering you on, and hopefully bouncing off ideas on you. And if as a result of this interview, anyone has questions, I would like to come back to you, and so that even in the in-between time, we can continue to learn from you and hopefully share insights with everyone.
Yes, and Auntie, if I can just say one thing, as you know, doing all these, you really need someone behind you, and your sister, and how much she and the girls have really given me the space, the support, and everything, and sometimes even the education, pointing out to me how I need to stay humble, and pointing out when I get the global awards, how I should put it into perspective, and their willingness to move with me, from Canada, Western Canada to Eastern Canada, to the States, multiple cities in the States, to China and back. It takes someone like your sister to be behind all this to enable it to work. So I just wanted to, put my hands out to her, and as you know her, she will continue to do sometimes even more than you and I put together.
Thank you. I admire you for acknowledging that your wife, Cynthia, is a major contributor to your success, and your 3 wonderful daughters who are also very successful in their own right, a topic for a different day. And it shows your humility, and I'm thankful that God has blessed you with such a wonderful family, and that He's using you to do all the fantastic things you're doing. So thank you for mentioning Cynthia, who is dear to me as well.
Thank you.
Definitely someone I admire also.
Okay. Thank you.
So thank you, and have a good evening And I'm sure we'll keep talking.
So thank you too, and thanks so much for giving me this opportunity. And I hope that we can continue actually to learn from one another all over the world, and that for those that are following us would know that it can be done. It can be done. I mean, when Obama said, yes, we can, it applies to every field. And we can also do it. Thank you.
I couldn't agree with you more. Thank you. Thank you for taking time to listen to this episode of RX How I Got There. This is a podcast where we bring you stories and insights from pharmacists to help you think more broadly about your own career. Please support us by subscribing to our channel, clicking like, and sharing this with your colleagues and your friends. Until the next time, this is your host, Edere Ibe.